while we have learning catalytics on, let me stop this one here and move on to the next one. All right, so this is just a review. Now, obviously, Dr. Taylor talked a lot about surface area to volume ratios as being like a major driver of form and function in animals. They're important in plants as well. And so we can think about two structures here, like a leaf and a tuber. And so, which of these is going to have a greater surface area to volume ratio? So is it the leaf or a tuber, which is a modified stem? There's an objective from lecture number 2 that actually think about, the main function of stems is to move the photosynthetic parts of the plant upright. But what are some functions of modified stems? And a tuber that's modified for storage is an example of that. All right, so let's see how we're doing here as far as the, okay. Looks like most people are able to, let's see how we're doing here. Our numbers look pretty good, so we're going to go ahead and keep going here with learning catalytics. So you have a few more seconds to answer this one. See if we can get some more responses. Most people are getting this one right here. All right, 5, 4, 3, 2, 1. Okay, good. So so about 87% got it right, that it's the leaf, right, that has greater surface area to volume ratio. And that makes sense, right? Because, the greater surface area to volume ratio you have, the more easy it is to have exchange with the environment. And so the leaf, we would need a lot of gas exchange happening here. Oxygen getting out, CO2 getting in. Now, of course, there's a penalty for that, right? Which means that you're also going to lose a lot of water through the leaf. So water loss, we talked about that in lecture number 1, is the flip side of that, having a lot of surface area for gas exchange. And then, of course, for harvesting light, you need a lot of surface area for that as well. So light absorption. But as it says here, the tuber is modified for storage, and its shape and reflects that, right? It's going as those cells in the center of the tuber are further away from the environment. There's going to be less loss of water and less exchange with the environment for that one. Yeah, so the surface area of the leaf. Oh, I should have mentioned, this is worksheet on page 7. I kind of jumping back to this one from the earlier one. So, all right. So keep your learning catalytics open for a moment. I'm going to go into some new material here, but then we're going to answer a few more questions, probably in 5 minutes or so, as we move on to the topic for this lecture, which is really thinking about water. So last time we thought about the challenges of water for animals, both animals in the ocean and freshwater, and a little bit about terrestrial animals. So today we're going to think about water. Plants have the same problem, that they have to get water into their bodies and they have to move it throughout their bodies. So you might have seen my little video here. Here's the tallest palm tree in San Luis Obispo. And the only way that the cells up here in the top of the plant can get water is from the roots down here. And so that water has to move a very long distance through the plant. So we'll think about how that works. Now, I'm going to build on some stuff that Dr. Taylor talked about, and I'm going to remind you, this might be familiar. Oh yeah, by the way, we're on worksheet page 11 for there. This should be familiar from a class like Bio 161, where you did this experiment with this little dialysis bag, which is basically, the dialysis bag is actually made of the same thing that plant cell walls are made out of, which is cellulose. It's just a thin sort of mesh of cellulose. And in Bio 161, you put a solution with a very high osmolarity inside the bag, right? So it had a high concentration of starch and sugar. And then you put that into a solution that had a very low osmolarity. Basically, it was just distilled water, right? And, you watched and measured this as water moved into the bag through osmosis, right? So not a big surprise there, right? The bag gains weight as the water moves in there. But now I'm going to ask you, okay, would there be a way to prevent any net movement of water, right? There's always going to be water moving back and forth, right? If we don't do anything, water is going to move into the bag. But is there a way that we can prevent any net movement of water in this system? Can we manipulate it in some way to prevent any net movement of water. Now, I know you're all looking at the worksheet, so you can kind of see the answer to this. I'm just going to jump ahead and do this fast. You can do that by squeezing the bag, right? You squeeze the bag hard enough, you're going to force water out, even though the osmotic gradient wants to have the water move in, right? And you could, by squeezing it just at the right amount of pressure, you could get to the point where you're balancing it off between the osmotic pull of water into the cell and the pressure squeezing water out of the cell, right? So you can find this balancing point where there's no net movement of water. Now, the reason I want to introduce this is because this idea of pressure is going to be very important for thinking about how water moves in plants. We have to think about solids still, just like we did with the animal lecture, but now we also have to think a lot about pressure, all right? And pressure is created by something that is unique to plant cells, at least different from plant cells and animal cells, which is the cell wall, right? So plant cells have this cell wall made of long strands of cellulose and a bunch of other important molecules. We'll look in more detail at the cell wall later. But right now, what you need to know is that the cell wall is this kind of semi-rigid structure, right? It's able to flex a little bit, but it can't change its shape or its size indefinitely. And so it puts constraints on the ability of the plant cell to swell up. So let's just kind of draw this together really quick here. So here's a cell, we have a nucleus, we can have some fun drawing some DNA in the nucleus here, blah, blah, blah. And then of course, we're also going to have lots of organelles. You know, we could draw some little mitochondria in here and maybe a chloroplast, since this is a plant cell. And then there's ribosomes and lots of proteins, and of course, lots and lots of ions and small molecules and things like that. So we have a very high concentration of solute here inside the cell, right? Because of all of the cell components. You know, like biophysical chemists tell us that there's about enough water in a cell to cover every molecule with like a monolayer, right? So think of all those proteins and everything inside of a typical eukaryotic cell. There's about a single layer of water separating each thing in there. So it's very, very densely packed. There is not much free water. So in other words, we have a very, very high osmolarity inside the cell. So here we'll imagine the cell in an environment where the solute or the osmolarity is much lower, you know, and this could be distilled water, obviously, but even if there's some solutes out there, there's still going to be this osmotic gradient into the cell. So water is going to, you know, and here's our plasma membrane. We could, you know, draw some integral membrane proteins here on the plasma membrane that are going to be pumping solute in, you know, to concentrate things here inside the cell. And water is going to move via osmosis into the cell from the environment. Okay. So now let's make this into a good plant cell. So we have to draw something in, obviously. So we're going to draw in the cell wall. So the cell wall is this layer of cellulose around the edge of the cell here. So we could draw some little strands of cellulose in here. And again, it's more complicated than just cellulose, and we'll get into some of the other molecules that live in the wall later. So here's our cell wall. Right? It's, I guess up here, it's semi-rigid, right? So it can stretch a little bit, but within, but there are some, it reaches its limit of expansion at some point. So semi-rigid, and yeah, it resists expansion. So what's going to happen here is that, you know, the volume, so water, you know, is being drawn into the cell osmotically, and it's going to create pressure pushing out here. So this is the, that arrow represents some pressure pushing out. And, you know, the cell's going to want to swell up, but eventually the cell wall is going to start to push back against that. And so there's going to be this equal and opposite pressure here. And that's where, you know, the net movement of water starts. So we call this the wall pressure. And then this is a really important term, this pressure pushing out from inside the cell. This is called the turgor pressure. Turgor pressure. And it's spelled up, you know, it's written out up there in the corner. You know, another way we'd say this is that a cell that is in a well-watered environment, at least, is turgid. just implying that it is under tension. Now, this is a pretty simple drawing of a plant cell. You know, like as you will read about, you know, I left an important organelle out of here, which is the vacuole. And there are some cells, some type of plants that use their vacuole in a very, in a way to really help them draw in a lot of water from the environment. So you read about plants that concentrate really high levels of salts, especially into their vacuoles, to help them pull water in from their environment. And it's especially important for those plants that live in very dry environments. But this is sort of a good starting point here. Okay, so yeah, that was what I wanted to say about the introduction here. So can I answer any questions, clarify anything on this drawing before we do some questions? Yeah. Oh yeah, so this is just, you know, so these are, I was trying to remind you that there's active transport going on, you know, and the cell is actively, you know, pumping new solute into the cell. And also it's, you know, creating proteins and things like that in the cytoplasm. So that's where a lot of those solids are coming from. Good. All right. Yes? So the pressure is from the inside of the cell pushing out. Yes, right. So yeah, so we've got the wall kind of resisting, and then this, you know, to anthropomorphize a little bit, right? The water wants to move into the cell because there's so much solid in there. But you know, if there were no cell wall, what would happen? The cell would just start to expand. That's what would happen with one of our cells if we put it into a environment where there was very low external osmolarity, right? It would start to swell up, and it might even burst if it swells up enough. But here, the cell wall is preventing that, and so the pressure that builds up is called trigger pressure. Yeah. Yeah, so, you know, we're anthropomorphizing a little bit. Yes, the water wants to move into the cell. It's better to say there is a higher concentration of free water outside the cell and a low concentration of free water inside the cell. So there's an osmotic gradient that's going to, you know, preferentially move water in. But eventually, the cell wall, the trigger pressure builds up, and then you stop any net water movement. Yeah, kind of, right. So yeah, that was just the hand was really just I would say it's kind of wrong that the cell wall is like, you know, like with a hand, you have this muscular pressure that you're squeezing on. I mean, that's how like our circulatory system works, right? That's the heart squeezes and pushes fluid around. Here, it's more, you know, the cell wall is inert, right? But it just resists expansion, and that's what causes the pressure. So let's try it. Let's move on here and try some questions. Now, these aren't in your notes or anything like that. I just kind of decided to add these here. Oops. Oh, I gotta stop. Sorry. All right, so you can start thinking about this. There we go. And I will start delivering this. Now, these are not plant cells, right? So imagine these little purple circles here is just animal cells, or a cell without a cell wall, right? And so we've got a couple different cells, and the osmolarity, or the concentration of solid inside and outside. And so the question is, in which scenario there would be the greatest net movement of water into the cell? So go ahead and think about this yourself, talk to people around you if you want to. This is not intended to be a very challenging question here, but it just gets you thinking about it. And the main thing about the numbers here is it just isn't really going to force you to kind of look at all the numbers and think about, okay, which direction is water moving in every scenario. Question? No, these are no cell walls or anything. These are just animal cells without walls. Oh yeah, the numbers are solute concentrations, yes. Let's say something like sucrose. If it helps you to have a. Okay, I don't want to go too long on this one because I have a feeling this one is a pretty low barrier question. All right. 5, 4, 3, 2, 1. Okay, good. So almost everybody got it right. If you didn't get it right, just make sure you are going to want to, sorry. You want to make sure you look at the numbers carefully. So D is the best answer here, where we have the biggest. So with the first two, you know, we're going to have water wanting to move out of the cell, right, from an area of low solid concentration, low osmolarity, to an area of higher osmolarity. And with the bottom two, it's D, where you've got the greater. I mean, you're going to have water moving in on both of those, but indeed, there's a greater difference in osmolarity between the outside and the inside. So D is the answer here. Okay, so now we're going to think about, with two of the cells, we're going to imagine these plant cells here, and the yellow represents a cell wall. So, so we have now two plants with a cell wall, and I'm going to ask you the, I'm going to ask you the same. You can start thinking about it, but the question's the same. Where is the net movement of water going to be the greatest? Into the cell. So go ahead and think about this yourself, work on it with people around you. And your answer will change this time. So. All right, good. We're getting. Let's go about 10 more seconds. Give everybody a chance here. Log in. All right, 5, 4, 3, 2, 1. Okay. So good. Like everybody is, almost everybody has moved to C as the correct answer right now. So just to look at this, you know, like our first two, that doesn't change, right? The cell wall doesn't change what's going on in B, because water is still going to want to move from inside the cell to outside. But now the answer has changed down here. Rather than me explain it, why don't you try to explain it yourself? You can do this, you know, in your notebook, if that's your style, or you can do it to somebody sitting next to you. But can you explain why the answer changed? And try and incorporate the word Turgor pressure into that answer, okay? So take a moment to do that. Why did the answer change from D to C in this case? And how is Turgor pressure a part of that answer? So maybe I'll change I'll pay a little more attention to the size of my arrows here, right? So in A and B, right, water is still going to be moving out of the cell. In D, now what we have is, you know, is that Turgor pressure will build up, right? So even though the osmotic gradient is greater, right, that cell is going to expand to this point where the Turgor pressure starts to exert sort of an equal and opposite pressure. The wall is going to exert an equal and opposite pressure. So we're going to have pressure pushing out from inside the cell. And eventually we're going to get to that point where there's no net change in water between them. And my point of showing you all this is that because of the cell wall, we got to think about pressure in plants. And sometimes pressure is going to win out, right? If we just looked at the numbers here, we would say one thing. But when we factor in the idea that there's a cell wall and that the cell is under pressure, you know, the magnitude of the solute difference is not the critical thing for determining which direction water is going to move here. And that's an important thing that you'll have to think about as you study this with plants. You can put your learning catalytics away now, because we're not going to do any more for, oops, till the Sorry, put the wrong thing there. Yeah, hold on. Okay, so we'll do some more later on here. Okay. So you can, oh yeah, question.
At what point, or if any, is the cell wall overcome by the pressure?
Right now, so the question is, at what point? Like, you know, like, oh, what exact solvent concentration is going to, like, that's a hard question for me to answer because different plant cells have different thicknesses of cell walls. The rigidity of the cell wall can be different. And you know, there are certainly ways we could calculate it. Like I could give you information about the wall, I could give you information about the osmotic gradient, and you could make a prediction of exactly where that would happen. I'm not going to ask you to do that math. We're going to think about all this stuff more qualitatively than quantitatively. So in here, the numbers are just sort of giving us a starting point. You could definitely calculate that, but you realize that, you know, knowing something about the makeup of the wall and how rigid it is going to influence that too. So, all right. Now you can go, so, you know, this idea of turgor pressure is really important for structuring plants. And you can go online and see tons of videos of people who have wilted plants that they're watering them, and then they, you know, sort of do a little time lapse of how they come back to life here. So I would encourage you to look at those for fun. Now, so for plants that are, you know, we would describe as herbaceous, meaning that they don't have a lot of woody structures, like this house plant here, you know, the turgor pressure is really what holds the plant upright. And so if the plant starts to die out, the water, you know, the cells are no longer turgid and the plant is going to wilt. But it can, you know, as long as it doesn't stay in that state for too long, it can rapidly be returned by watering it. I thought this quote was really neat. Plants are made of tiny pressure bombs. Turgor pressure can reach 20 atmospheres or 2 millipascals, a value much higher than the air pressure inside automobile tires. So keep that in mind, right? So in a well-watered plant, those tiny little cells are under, there is a lot of pressure, right, in those cells. And just something that you can kind of imagine here is if we took our cell here that's turgid, just think about this for a second. If we were to poke a hole through that cell wall, then what's going to happen, right? We have all this pressure like pushing out from inside, that turgor pressure. If we poked a hole in it, fluid is going to start to move out of that cell, right? That just sort of intuitively hopefully makes sense. If it's a little pressure bomb and we poke a hole in it, what's going to happen, right? That fluid is going to exit. So keep that in mind for later. That's going to be helpful. But I think it's useful to remember, right, that plant cells, even though it's on a very, very tiny scale, they are under very, very high pressure. Okay. So now here's where I have to take something that is, frankly, you know, kind of an easy thing to think about intuitively, and make it kind of complicated by adding a lot of vocabulary to it. And the reason is because as I've established so far, when you're dealing with plants and you're thinking about water moving through the plant, you have to think about solute, and you also have to think about pressure. And so plant biologists have decided that it would be useful to have something that combines both of the two. And so I'm going to introduce you to this new idea called water potential, and it's represented by the Greek symbol psi. So that represents the water potential of a particular volume. And the definition that you'll see in the book is water potential is the potential energy water has in a particular environment. Okay, potential energy, I know what that is, right? That's like water, that's like, you know, a battery or something like that, or a chemical bond like in an ATP or something. That has high potential energy. I kind of understand that concept, but what does that mean in terms of water? So below that in parentheses is how I like to think of it, and I think this is probably helpful too. It's the likelihood that water will move away from a cell or a region of an organism. So if water potential is high, that means the water wants to move somewhere else. If water potential is low, that means water is going to tend to move into that area. Okay, so high water potential means the water wants to move away somewhere else. Now, as I said, there's two things that will affect water potential. And over here, all of the things on this side have high water potential. And all the things over on this side here have lower water potential. And in every case, the water is going to move in that direction. So in the top is solid, and that's probably, you know, that's familiar from the animal lecture and from other things, right? Water is going to move from an area of low solid into an area of higher solid, right? But now in the lower two, we're going to think about pressure. And for pressure, there's actually sort of two ways we can think about it. We can think about the fire hydrant here exerting positive pressure. Right? And so the water is going to want to move away from that source of high positive pressure. And now in the lower scenario here with the juice box, we've got sort of the opposite thing, where by sucking on the straw, I mean, there's, by sucking on the straw, you're creating an area of low pressure up at the top of that straw. And that is also going to potentially, positive pressure, I guess not low pressure, I'm going to say negative pressure. Also cause fluid to move, right? So in every case here, right, so everything over in this first column, those are examples of things where the water potential is high, either because of solid or because of of pressure. And then over here we have situations where we have a lower water potential, and in every case the water is going to move from high to low. So keep that in mind, right? Water always moves, the flow of water is always from a region of high water potential to a region of low water potential. Now, well, actually, I'll go on to the next thing here. So we're going to think a little bit more detail about the effects of solute and pressure here. So here's the equation, right? It's really simple. The water potential just equals these two. You have to add together these two things. And psi sub s, that is the solute potential, right? So in other words, that's the effect of solute has on this particular volume of water. And then psi sub p, that is the pressure potential. So remember, the whole point of doing this is because we want to be able to sort of combine these two things, solute and pressure, together into one number that will give us some information about where water is going to move. Okay, here's where things get a little bit more complicated. So let's just look at the solute potential. This is the contribution of solute to the movement of water. It is always negative or zero. If you have like basically pure water, water with no, just pure H2O with not a single dissolved molecule in it, zero solute, then the solute potential is zero. So pure water. And that kind of makes sense. You can't have like negative solute, right? You can't have a negative solute concentration, right? You can have no solute, but you can't have negative solute. So as soon as you start adding solute to a solution, then that solute potential is going to get lower and lower and lower. And this is hard for students in this class because this is the opposite of osmolarity, right? With osmolarity, the more solute you add, the higher the osmolarity goes. Here, and this is just something you have to get used to thinking about, as you add solute, this value, at least the solute potential, is going to get lower and lower and lower. So a really, really salty solution is going to have a very low solute potential. And it kind of makes, water is going to want, if you have a really salty solution and there's water around it, it's going to want to move into that region. And we're thinking about, describing how water is going to move here. So it's inverse to the concentration of solute. Pressure is a little bit easier to think about. Okay, it can be positive or negative. So you can, by squeezing a volume, by squeezing something, you're creating positive pressure. Like by sucking on the end of the straw, you're creating negative pressure. So this could be either positive or negative. And it's a direct relationship with pressure. So this, the psi sub p, the pressure potential, goes up and down as the pressure goes up and down. It could also be zero, of course, too, if there's no pressure, positive or negative pressure. And then we add those two together, and we get the overall water potential. So we're going to combine these two, think about the contribution. I mean, we're always interested, which way is water going to move? And we got to think about the contribution of solute and the contribution of pressure. Now, when you read in the textbook here on these pages, it actually goes through some calculations for this. And I think that's useful to do. I'm not going to ask you to do that. We're all, like I said before, we're going to think about this qualitatively and not quantitatively. But I think doing the quantitative questions, I think there maybe is like one quantitative question in the homework as well. It's going to help you kind of learn this. Okay, now we're going to start to apply these later. So let's just, I wanted to establish the definitions here, and now we're going to go on to think about these. But here's one more thing I want to leave you with, and here we're on page 13 of the workbook. What I'm going to say here, this is going to be definitely true of plants. And I don't know of any exceptions to this in animals. So I think you can always kind of think about this as you're thinking about fluid moving through the body. Short distance movement. So short distance movement, when we think about like into or out of a cell, or maybe from the, from the, the loop of Henle into the surrounding interstitial fluid or something like that. These short distance movements is almost always driven by solute. That's going to be the dominant thing for these short distance movements. However, long distance movements, from one tissue or region to another, is almost always driven by pressure. All right. So I think that makes sense. So moving water into or out of a cell, that's going to be primarily due to solute differences. Moving water from a root to a leaf is primarily going to be due to pressure. And I think it's the same for animals, right? We will learn about the circulatory system in the next lecture, but of course the heart, it is a muscle that puts pressure on fluids in there, and that forces, that pressure is what forces the fluid through your circulatory system. So these are general statements that will be helpful for you as you think about both plant and animal water movement. All right. So I just said that, right? So long distance, almost always due to pressure. So now let's think about, here's long distance water movement from the roots to the leaves in a tall tree, all right? Which do you think is most important? Now, this is not a graded question or anything like that. It's not learning catalytics. Just which do you think? Do you think it's positive pressure, water being pushed up from the roots up to the leaves? Or is it negative pressure? Are the leaves somehow like sucking the water up from the bottom? Which do you think is most important? So go ahead and just think about this for a moment. Take your best guess, and we'll come back and we'll answer the question later. Which do you think is the most important? Positive pressure from the root or negative pressure created up in the leaves? All right. So I'll tell you, it turns out both of these contribute. But one has a greater contribution than the other. So let's think about it. All right, so now we're going to follow the movement of water. This sort of, it's been known for a long time that water has to move from the roots up to the leaves. But in terms of figuring out how that worked, is still, there's still parts of the process that are under investigation, right? So we're going to start off here in the soil, where of course we have water in the soil. And then here we have the root. Let's draw a root meristem down here. And then we have some cells that are starting to differentiate. We have some epidermal cells. We have other types of cells. And then above that, this is vascular cells. So little older cells. This is the vascular tissue called the xylem. By the way, this was introduced in chapter 34, and there were some objectives about xylem and phloem. We're going to look at much more detail at that now. Okay, so the solute concentration, this is going to sound a little weird, but the solute concentration in soil is very, very low. Low solute, low osmolarity. Soil water is actually very, very dilute. Which seems kind of weird because at our macro level, when we think about soil, think about like a mud puddle or something like that. You feel like, oh, there's a lot of stuff in there, But at the macro level, at the micro level, the water that's in soil is very dilute. So soil is made up of, it's a complex thing, but it's like small pieces of sand, small pieces of clay, small rocks, things like that. Of course, there are some dissolved nutrients in the water, but really, typically what you have in soil is they've got these small particles that have a little shell of water around them, and that water is very dilute. So even though our macro view of soil might tell us something different, just remember that soil water is very dilute. Now, meanwhile, here inside these root cells, I guess I'll just draw it out here, we have a much higher concentration of solute. Some of that is sugars that have been brought down from the top part of the plant, but it's also all those things we talked about before, the proteins and small molecules inside those cells. And so the movement of water here from the soil into the root is osmotic, right? So this is really driven by, I'll say driven by solute. So this is very similar to the picture of the cell we drew earlier, where we've got the cell with a high concentration of solute inside, and it's in a dilute environment. So what's going to happen here? Water is going to move into the root, into those cells. They're going to start to swell up, but then that's where pressure kicks in. Because remember, they've got cell walls. And so they're going to start to swell up, and the pressure is going to build up. And if there wasn't like a way for that pressure to escape, that would be, yeah, we would just sort of reach equilibrium and there'd be no net movement of water. But above these cells here in the root, there is sort of an escape hatch for that pressure, and it is these xylem cells. And so water is going to get forced up into the xylem from these cells here, and that is driven by pressure, right? So, so let's think again what's happening here, right? The dilute water in the environment is going to be brought into those cells through osmosis. Those cells are going to start to swell up, trigger pressure builds up in those cells, and that's going to force the water up into the xylem. And so this is driven by pressure. Driven by, and this, I'm going to say positive pressure, right? Because it's really, you know, essentially those cells in the root sort of pushing the water up, right? They're squeezing the water up into the xylem. All right, now we need to think a little bit about xylem cells, and you looked at some of these in lab as well. So remember, xylem, this is an example of a cell that is dead at functional maturity. I like that term, dead at functional maturity. So these cells, of course, while they're born inside of a meristem, just like any cell in the plant would be, and then they start to grow, they start to elongate, they secrete their cell walls, and then eventually, once they have this elongated shape, they undergo programmed cell death. And so the cell dies, its nutrients are used by other parts of the plant, and what's left over is the cell wall. And while they're growing, a few important, so this is just the wall here of the xylem. And a few important things happen. First of all, there are connections established between adjacent cells. So this is, this little plate here. In my drawing, there were 1, 2, 3 cells that are now gone, and their cell walls are all that are left there. And so these are called perforations down here. Perforation. And these are just holes in that cell wall that are going to make it easier for water to move up through this column, right? So now as these cells are growing, they also have some regions, sometimes water has to get out of the xylem. And so there's little holes around here called pits. And again, these are formed when the cell is alive. And then when it dies, all that's left over is that cell wall. And so these pits are going to allow water to move out of the xylem at some points, and so and move into the xylem at other points. And so essentially we have this hollow tube of cell wall that is, those cells have created this structure that is sort of ideal for moving water long distances, and the root cells are going to be forcing water up into that column. All right, see people drawing away there. So let me give you some time to catch up, and let me answer any questions or clarify anything if you have any questions. Okay, this is something as you study this, I would encourage you to replay it in your mind. Okay, high concentration of solute in the root cells that draws water in from the environment where the water is much more dilute. Trigger pressure builds up in those cells, and there's an escape hatch for that pressure, which is up into the xylem. So we have positive pressure in the root cells that's pushing water up into the xylem. All right, this is called, so what we're seeing here is positive pressure from the root. And really, this accounts for movement of water up the plant somewhat, but by most people's estimates, this will only take water, there's only enough positive pressure from the root to push the water up maybe a meter or so up a plant. So for a very small plant, this would be enough. But for a big tree or that palm tree downtown, this isn't enough. So really only maybe for, 1 to 2 meters up the stem. But there's a lot of plants that are much, much bigger than that. So this gets us started with this. Yes.
So what role does its play exactly?
Oh, again, so the, oh, you know, I guess, hold on, let me show you the next slide, then come back to answer your question. Hang on just a second here. You don't have this in your notebook, and this, because I've got a bunch of questions about it in office hours. I said something about lignin in one of my objectives. I think it's in the lecture 2 objectives. This is a picture of lignin. You do not need to know this, but lignin is something that is synthesized in these cells as they're developing, and they secrete it out into their cell walls. And it has two it has two effects on those cell walls. First of all, it makes them more rigid, but also this molecule is much more hydrophobic than cellulose. And so this makes the cell walls somewhat hydrophobic, and that makes them better at conducting water, right? Because, you know, if it was just cellulose, it'd be like a paper towel. Water can kind of go in and out through the cellulose, but with this lignin in there, it makes them more hydrophobic. And so if you imagine that the cell walls of the xylem, here's to answer your question, the cell walls of the xylem have this sort of hydrophobic quality, then it does actually help to have some little holes in there to let water escape. Okay. All right. Hey, thank you for waiting over here. What was your question?
If the pressure becomes high enough to burst the xylem.
Oh, would the pressure become high enough to burst the xylem? No, I don't think so, because first of all, the xylem cells are pretty rigid, you know, so like you could, I suppose you could imagine a point where like there's, you know, only so much water that's going to get forced up there, but they're, because of the lignin, again, they have quite a bit of rigidity, so they're not going to like swell up and rupture or anything like that. There was another, yes.
Would plasmodesmata be present in xylem cells?
Good question. Would plasmodesmata be present in xylem cells? I think we're going to, that term will come up later, but I would refer to that, like, I mean, like these perforations, you're probably looking at those and thinking that sounds a lot like plasmodesmata. These are actually quite a bit larger than plasmodesmata, which really, plasmodesmata are cytoplasmic connections between living cells. So I would use that definition exclusively for that. And we will see an example later, but these are big openings that just let the water transport more fully. Yeah. Oh yeah, right. Dead at functional maturity. This is a term that I want you to know well, right? And what that means, functional. So if you think about a cell that's mature, that means that it has gone from an undifferentiated cell in a meristem, and it has, you know, through gene expression and, you know, making a lot of different proteins and making a lot of different structures, it has assumed a particular shape and structure that is important for it to do its function, right? And so an epidermal cell is going to have a different, at maturity, is going to look different than a guard cell, which like on the border of a stomata, which is going to be different than a mesophyll cell, which is going to be different than a xylem cell. So guard cells, epidermal cells, mesophyll cells, those are all alive at functional maturity. They have to be doing all the stuff a living cell does. They're doing photosynthesis, they're, you know, doing active transport and those sort of things. Xylem, on the other hand, when it is ready to do the job that it was born to do, it has got to be dead to do that job, right? So a living xylem cell is not any use to the plant. So at functional maturity, when it reaches that state where it's ready to do its job, the cell is actually, the death of the cell is the last part of that. Yeah. Yeah, so that's, you know, that little disc in there, that would be described, I think, as the plate between the two cells. And so, yeah, remember, this drawing, at one point, if we roll the time back, this was 3 cells that were all turning into xylem. You know, they were on their way to becoming xylem cells. They formed the cell walls, they formed the pits and the perforations, and then now the cytoplasm is just gone. Yeah.
Is the reason we have like dead functional cells just as like agents for like resources and energy, or?
Right. So the question is about, okay, having these dead cells. I think, you know, plants have this challenge that they have to move water long distances. And in this case, you know, the evolutionary solution was these cells that just die and leave their, you know, these sort of hydrophobic tubes running up and down through the plant. That was the most efficient. You couldn't imagine a living cell that could move water. And in fact, you know, for the other part of the vascular system, the phloem, those cells are all alive and they have to be alive. But for this, the evolutionary solution of moving water up the plant from the roots to the leaves, it was, it's these dead cells that do it. All right. So, hey, look, we've got the water into the root and we've got it up maybe a meter or two in the plant, but we still have a long way to go, right? There's some big trees out there. So let's think about the rest of it. So here we have to get to what's called the cohesion tension theory that explains the rest of the water motion. Now, this is a very simple drawing of a plant, obviously. Here's a leaf, we have the stem, we have the root down here. So we could just really quickly recapitulate what we wrote before here. We had water that goes into the root, and then we have a little positive pressure that pushes that water up here into the lower part of the stem, right? So that's that whole previous slide. So that water is in the xylem. We're not going to draw the xylem cells. But it still has to go a long way here up into the leaf. And so inside the leaf, remember, we have mesophyll cells. And we have a lot of air space in there as well, right? And so something we've talked about as a challenge for plants during photosynthesis is transpiration, right? The loss of water through these cells. So remember here, you can see on the bottom of this, we have some stomata that are open there. And so water is going to move off of these cells into, well, here, hold on, I'll maybe draw one water molecule here in the airspace, and then that's going to move out here into the environment. Okay. So here's what's kind of amazing is that, there is an unbroken chain of water molecules. Now, of course, these are inside these xylem cells that run up and they grow into the leaves and everything like that. And then they terminate here inside the leaf by these mesophyll cells. And that's where the water is coming out. But as this water molecule leaves, so this is transpiration. As this water molecule leaves the moist surface of those mesophyll cells, it's going to just pull a little bit on the water molecules behind it. And it does so because water molecules are polar and they make hydrogen bonds with each other. So we got to think about hydrogen bonding. And in fact, that's the cohesion part of this is hydrogen bonds between water molecules. And so this hydrogen, I'll use dotted lines to represent the hydrogen bonds, and so we've got hydrogen bonds connecting all of these little water molecules in this column of water that extends from the root all the way up there. And as that water molecule transpires away, as it leaves the surface and goes off into the environment, it just exerts this tiny little pull on the water molecules behind it because of hydrogen bonding. And then those molecules exert a little pull on the water behind them, and then they pull on the one behind them. And believe it or not, this tiny, tiny little force is enough to move water, up many meters. There's trees that are 100 meters tall, right? And these tiny little forces are enough to move water most of this way. And here's where like pressure and things like that come into it. This transpiration is a source of negative pressure. So I'll say, creates negative pressure in the leaf. I forgot to bring my own box of Kleenex, so at least nobody tell anything weird, but I stole these from backstage. So this is a good sort of representation of it. So here we have this Kleenex sticking out of the top, and there's a bunch of Kleenexes in the box, right? And they're not attached to each other, they're kind of wrapped around each other, right? And so you can imagine this as a water molecule here. And if this is going to work right? So the sun shines down on this leaf, this water molecule goes off into the environment, and as it did so, it pulled a little bit on the one behind it, right? And so that one popped up, and now as we do this, each one of these is going to exert this little pull. Now imagine a box of Kleenex that was like 100 meters tall, right? And the Kleenexes are all wrapped around each other all the way down to the bottom. That would be the equivalent of this column of water, and so that little tiny force of those water molecules transpiring off the top are pulling on everything else. So the tension part of the cohesion tension theory that explains this long distance movement of water is, I'll just write this out as water transpires. It pulls on the water column. Now, another word that you'll read about in the objectives in the chapter is adhesion. It sounds a lot like cohesion, and it's also involved in hydrogen bonding. But so cohesion talks about water molecules hydrogen bonding with each other, which is the critical thing I want you to think about here. Adhesion is the water molecules making hydrogen bonds to the cell walls around of the xylem. So in addition, that's kind of what prevents, like you could say, oh, if transpiration stops, why doesn't the water just like drain out of the plant? Well, there's a couple of things preventing that. One is that the water molecules are also sticking to the cell walls around them in addition to sticking to each other. And then also remember, you have this little bit of positive pressure pushing up from the root that is going to help support that whole water column as well. So at night, where there's very little transpiration happening, the water doesn't just like drain out of the plant. All right. So I have a good way of summarizing this here. Oh yeah, just to remind you, right? So here's stomata. Here's the airspace inside the leaf that is the source of this transpiration. And then here you have this on the top of page 15, if you have the printed version of the workbook. And this is just a real good way for us to summarize this quickly. So we are getting water moving from the soil to the root, to the stem, to the leaf, and to the air. And I wanted to take a moment to go back to those terms I introduced earlier, which is water potential. So water always moves from an area of high water potential to an area of low water potential. And so the highest water potential must be here in the soil. Whereas the lowest water potential is here in the air. And at each step, the water potential is going to get lower and lower and lower. Water always is going to want to move from an area of high water potential into an area of low water potential. Now, what is the driving force at each of these points? That is different. So here, from the soil to the root, this is solute driven. So solute is the main driver of water in that part. There isn't really a pressure component, right? It would be wrong to think, oh, is the soil like squeezing the water into the plant? That's not really what's happening, right? There isn't a pressure component. This is solute driven. From the root up into at least the lower part of the stem, that is positive pressure from the root that's creating that. So positive pressure from the root. And then the rest of it here, which is for of our big tree, is most of the distance. This is created by negative pressure in the leaves. And that is really due to transpiration, right? Those water molecules leaving, creating this area of low pressure up inside the leaf, right? As it moves from the surface of those leaves off into the airspace in the leaf and then eventually into the environment. So solute and pressure both play a role, but for the, to go back to this, I'll come back to this in a second, but for this question here, right? For most big plants, the negative pressure created up in the leaf is the primary driver of water up and out of the plant. All right. Give everybody a moment to. Now we're going to shift our focus and think about water moving the other way, right? Because we also have to, those root cells down there, they need sugar. The only place to make sugar in the plant is up in the leaves. So you have to think about water going the other way as well and carrying not just sugar, but other molecules in it. So that's going to come up next here. All right, but this gives us our idea of moving water from the roots up from the soil into the plant and up to the leaves. All right. So now we're going to shift our attention to phloem cells. So this is our other major component of the vascular system. And we've done already a little bit of reading and studying about this. So we've got two types of cells that make up the phloem. They are the sieve tubes and the companion cells. And so here we have a little comparison for them. These cells are all alive at functional maturity. So everything we're going to talk about in the phloem, these are all living cells. and live like all living cells, they have a cytoplasm. So both the companion cell and the sieve tube cell, they both have a cytoplasm. So we could draw some little, proteins and things like that in here. And they both have a plasma membrane. So even though it's, what we're mostly in the illustration above, the shapes you see are really representing the cell walls, but they have a plasma membrane. But now there's some differences that pop up. Only the companion cell has a nucleus, right? So only the companion cell does gene expression. Mitochondria also are only found, as far as we know, in the companion cell. So it's going to be the main source of ATP. And also protein synthesis seems to only happen in the companion cell. So again, gene expression, protein synthesis, right, is going to happen here. Now here, the term plasmodesmata is appropriate to use because these cells, they do have that linked cytoplasm. They're sharing their cytoplasm via plasmodesmata. And so the sieve tube, the companion cell could really survive on its own, right? It has all the things it needs to survive, but the sieve tube cell, it can't. It is really just essentially an empty cytoplasm, right? There are some proteins and things like that in there, but it is it's just an empty cytoplasm. And the sieve tube cells, as they grow and develop, they form something kind of like the perforations that we saw in the xylem, but there are large openings between these sieve tube cells, which are going to make the movement of fluid much more efficient. Just breaks in the cell wall there that allow that to happen. Okay, so those are our two key players. We got the sieve tube cells and the companion cells. And now another set of vocabulary that we need to think about for talking about movement in the phloem is we're going to describe tissues as either a source or a sink. With the xylem, it's a lot easier because we're always moving, the water has to always move in the direction of where transpiration is taking place. We can't move it in any other direction because basically transpiration is what is driving the whole thing. But with the phloem, we have to move things around different directions of the plant. So we're going to talk about always moving via the phloem. Translocation is always going to move from a tissue called a source to a sink. So this is called translocation. Moving from a source to a sink. Now, so a source is a net exporter of sugar. In other words, it sends more sugar out to other parts of the plant than it consumes itself. Whereas a sink is a net consumer of sugar, right? So it's going to consume more than it makes. All right. So what are some examples here? Well, a leaf, if I asked you where does a leaf belong, you'd say, oh, of course it's a source. That's where photosynthesis is happening. But we have to kind of be careful, right? A young leaf that's fully photosynthetically active might be a source, but an older leaf or a leaf that is shaded by other leaves, those could actually become a sink. They start to consume more sugars than they eat or than they produce. And so they're going to receive sugars from other parts of the plant. Storage organ, like a tuber or something like that, when it's mature, it could be a source. That's a place where the plant has stored up, sugars in the form of starch usually, and then at some point in the plant, that starch is going to be mobilized and exported to other parts of the plant. Whereas where that same storage organ is developing, where it's growing, it might be accumulating sugars, right? It's going to be taking in sugars from leaves. I was going to have you think about this, but seeds, again, could go in either category. A seed during germination is a source of food for that little developing plant until it can start to photosynthesize on its own. Whereas a seed that is developing right after fertilization, so post-fertilization, that seed might be storing up sugars, as it's developing. So again, the source-sink relationship is you can't just easily put things into one category or the other. But in every case, we have to be able to move sugars from the source to the sink. And it does so via the phloem. All right, that's the tissue that does this. Okay, So here's a really important figure that we're gonna draw a lot on this, but don't draw this right now. Leave it blank right now. Just look at the screen and just think about this. Don't draw anything onto your figure yet, because we're gonna need the space. But okay, you can draw this. Up here, we're gonna say like, okay, this is a source up here at the top of this column, and down here, we're moving towards a sink. And the blue arrow, that just shows the direction that the fluid is gonna flow, right? It's gonna flow from the source to the sink. So let's try and apply this water potential idea here. Where is water potential going to be the highest? Is it going to be higher at the source or at the sink? So just giving you some opportunities to practice thinking about water potential here. We know the direction, right? The direction is moving from the source to the sink. So where is water potential the highest? We can just do this with hand signals. If you think it's higher in the source, do thumbs up. If you think it's higher in the sink, do thumbs down. Okay. Yeah. See mostly people going thumbs up. That's right. So we've got high water potential up here. So we could just say high water potential, and in the sink, we're going to have low water potential. Okay. So how does, now we have to think about, okay, what creates that? Let me, how is that high water potential created here in the sink, in the source, sorry, and how is the low water potential created in the sink? Okay. So these are the sieve tube cells that we've drawn in here. So we've got to draw in some other cells. And right now I'm going to ask you to kind of leave this part blank. We're going to draw something in there later. So just kind of draw along with me here as best you can. So we're going to draw a companion cell. Now, every one of these sieve tube cells, we've got 4 sieve tube cells here. Each one of them would have a companion cell, but we're only going to draw in 2 of them just to keep it a little bit simpler. We can draw in some little plasmodesmata to connect the cytoplasms of these guys. And we'll just take the easiest example of a source, which would be like a young leaf that's really photosynthetically active. So let's put over here next to this a mesophyll cell. So we've got, here's our mesophyll. Here's our companion cell. And a really good example of a sink, of course, would be a root cell, right? So the root cells, they need the sugars that the leaves are creating, but they can't photosynthesize on their own. So let's just draw a simple root cell down here as well. So here's a cell in the root. All right. So somehow we have to drive fluid from that source down towards the sink. Now, here in the mesophyll cell, we could do a quick little review of photosynthesis. There's carbon dioxide in the atmosphere. Through the Calvin cycle, that's going to be taken in and made into sugar. So just keep that really simple. That sugar then is going to be moved here into the companion cell. And if you think about this, a sugar molecule, it can't move by itself through a membrane, right? It's too big and it's polar to move by itself. So it is going to be moved through, some form of active transport. I'm just going to label this as active here, right? There are sugar pumps and channels and things like that that are going to move this sugar across, and we're going to end up with a high concentration of sugar because we're concentrating it through active transport. We're going to end up with this high concentration of sugar in these cells up here in the source, right? We're making lots of sugar, we're pumping it in to first into the companion cell, and then it diffuses over into the sieve tube cell. We're going to end up with this high concentration of sugar. All right, now let's change our attention down to the other end. So down here in the root, if a root cell gets a sugar molecule, what is it going to do? It is going to use it for glycolysis and citric acid and electron transport, and it is going to break that sugar molecule down, and it's going to release carbon dioxide, right? And so where's that sugar coming from? Well, it's getting pumped out of the companion cell here. And the result is that we're going to end up with a low concentration of sugar here we go. Down here near the sink. And again, I could draw some proteins, some integral membrane proteins here that are doing this. Is an active process of moving the sugar out of one cell and into another cell. Okay. So we're just getting it set up right now. So now we got to start thinking about pressure, because again, this is a long example. I remember a slide I showed you earlier. Long distance transport is almost always due to pressure. It's always the driving force. So we have to think about how pressure is going to work here, because this is definitely an example of long distance transport. All right, let's just keep this really simple right now. Let's put some water in the environment around these cells. All right, so here's a water molecule. As we increase the concentration of sugar inside those inside the phloem, water is going to move in there, right? That is just through osmosis. So we're going to get water moving into these cells because they have a high concentration of solid. So this is just movement by osmosis, short distance movement from the environment into those cells because of the high concentration of solid that's there. Now this always kind of bugs students when I draw it this way, but I'm trying to show something that is really complicated and three-dimensional, and I'm trying to keep it really simple and showing it two-dimensionally like this. But down here at the bottom, as we're taking sugar out of those cells, water is also going to flow out. Now, this is what bugs students. It's they're like, oh, but you showed the sugar going that way and the water going this way. Remember, this is three-dimensional, right? So the sugar is going to be going out in all directions and the water is going to be going out in all directions. Okay, but we got to keep it simple here. So here's where we got to think about pressure and sugar pressure. So up there at the source, you got a high concentration of solid. Water is going to start to be drawn into that cell. The cell is going to start to swell up, but the cell wall is going to start pushing back. So we've got a lot of sugar pressure. Remember, each of these cells is like a little pressure bomb, right? There's really high pressure there. In this case, there is an opening underneath, right? So down here, what we have is very high pressure. I'm sorry, up here we have high pressure is created. Down here at the bottom, we have lower pressure. And that is really the thing that's driving the water movement. So we're going to get water, we could call it sap if we wanted to. It's basically fluid with a lot of dissolved molecules, dissolved sugar in here, moving in this direction. So again, you got to think about, okay, there's some short distance water movement. We've got a lot of solute in those cells at the source. Water gets drawn in, pressure builds up. And in this case, there's a lower pressure region down here at the bottom, right? So we have low pressure because at the bottom, we're taking the solute out of those cells. The water is also following the solute out of the cells. And that's creating an area of lower pressure. So that's going to always have this movement going down. Now, I think some students, when they start looking at this, they get a little confused because they're like, wait a second, there's a high concentration of solutes up at the top and a low concentrations of solutes at the bottom. Shouldn't water be moving the other way? Doesn't water like moving from an area of low concentration of solute to an area of high concentration of solute? Yes, that's totally correct. But remember, in plants, you got to factor in pressure as well. So this is a case where, if we just looked at the concentration of sugar, we might predict that the movement of water would be that direction. But pressure is the dominant factor here. And so that, in this case, the pressure is working against the sugar concentrations and moving water downwards. Okay, so here's the last thing we have to add in. Where is what's going on with this water? Well, when you look in leaves, the xylem and the phloem are always right next to each other. So here we have, let's draw in some xylem with pits and perforations and all that kind of stuff, these xylem cells that we had before. So remember, there's this movement of water up the xylem here, and this water that is kind of, that is the source of this water that is coming into the cell up here at the top, right? So we've got, it's the way I've drawn it, it really does look like the water is sort of cycling. But just keep in mind the xylem and the phloem cells are right next to each other. They're closely connected. There's water moving up through the xylem because of the transpiration happening, and some of that water is able to get out, and that is the water that moves into the phloem. All right. So now what we have here is a very complicated diagram, right? That if you go back and look at this in your notes, you can stare at this diagram as it's written out right here for hours and not know how to do this. What you really need to do is draw this a bunch of times, right? And think about, okay, here's what's happening at the source, and that, you know, like kind of what I did. And so maybe having the recording of the lecture to watch is going to be helpful for that as well. So I did have some, you know, like some learning catalytics questions to follow up with this. But maybe I'll do that as like I've done on some of the other ones, is like sort of an optional little way of testing yourself with a little quiz or something like that, an ungraded quiz up on campus. But we have to stop right now. So I can hang out for a little bit. Sorry, I have time for questions right here at the end. But answer some questions before you take off. I'll see you back here on Wednesday. Yeah. So, remember, yeah. So the cellular, like I just say in the phloem, you know, the lignin in there does tend to be kind of hydrophobic, and that helps prolong the transport. The cells of the cell walls of the phloem don't have that same sort of hydrophobic quality, so it's just going to move through osmosis through the cellulose. Yeah, sure. Hi.
I was trying to finish my mastering bio
Yeah.
assignment last